Well, good morning, everyone. We are here today to attend the doctoral meeting of Noel Suárez Barro, who is a PhD student here in the CiTIUS. And the topic of this the doctoral meeting is From Molecules to Medicine, Why Toxicity Matters in Drug Discovery. This is a very interesting topic, even for those who are not related to the field, as myself, for example. And it's interesting because advances in medicine are among the most noteworthy advances in humanity, and the advances with greatest return. Indeed, one just need to think about the life expectancy. For example, the life expectancy was around 30 years from the Bronze Age, from the records that we got, until something like the Industrial Revolution. And in the Industrial Revolution, the life expectancy grew above 40 years. And it wasn't until past the World War II when the life expectancy grew about the 70, 80 years of lifespan. And this life expectancy grew just because of the advances in medicine, where vaccines and antibiotics were the spearheads of this research. And so we all here work in tech development, in developing new technologies, from conceiving, developing, and releasing technology. So we all know that this is a very long process, a very hard process. And indeed, drug discovery is not away from these problems. On the opposite, probably they are more challenging. So it has several processes, such as preclinical research, several phases of trial in human control groups, and all the regulatory needed and so on. One simply has to remember the most critical medical alert of our times, so long, that is the COVID. And with all the efforts of humanity focused on developing a vaccine, all the efforts of all the countries, and the time to develop such a drug was around 1 year. And that was completely astonishing in our times. But such is the problems that this topic focuses. And also, you all know here about the potential of AI in this to develop new technologies. And without further dilation, I give the word to Noel to present us his research. Thank you.
Well, thank you for the introduction. Today, I'm going to talk about how we go from molecules to medicines and why toxicity matters in drug discovery. Drug discovery is the process in which we try to develop new medicines to fix certain issues. We can divide it in 4 stages. First, we need to identify the cause of the problem that we want to solve. Then we need to find a molecule that fixes that problem and serve us as a cure to that disease, for example. Then we need to check that molecule can be used safely as a drug and has no adverse effects on the patient. And then we can commercialize the drug and take it to the market so the patients can use it, start using it. First, I'm going to do a little biological introduction to understand better this topic. Our body is mainly made up of proteins. Proteins are like the bricks of the body, the building blocks of the body. We can find them everywhere, in every tissue, with different functionalities. For example, we can move thanks to the proteins in our muscles. They can also make us think, because there are proteins involved in the transmission of the neural impulse. Also, our immune system works thanks to proteins, like antibodies that defend us and protect us from external pathogens that can be dangerous to us. And even in our blood, we can find hemoglobin, which is a protein responsible for carrying the oxygen to every part of our body, so we can live. One important thing of proteins is their structure. It is usually very relevant for their function and their working. And if this structure is lost, it can lose its functionality and not work properly. Its structure can also change during its functioning, its normal functioning. For example, here we see the hemoglobin changing structure depending on if it has oxygen attached or not. Most diseases are related to a dysfunction in some protein of our body. Maybe because it has lost its structure, or maybe because we have a genetic problem in which we are not able to codify that protein. In drug design, well, when we want to design a medicine or a farm, we usually care about this protein structure and try to find a small molecule that interacts in the way that we want with this protein, usually from a unknown structure, in order to obtain the result that we want and fix the problem that we may have. Another important issue in drug development is what we call the ADMET properties. ADMET are the letters in this acronym refer to these different properties that are desirable for a molecule to be used as a drug. We have A, that stands for absorption. This refers to how the medicine will get into our body. For example, by ingesting the drug, it reaches our stomach, and from then it can go to our blood. We can also inject it directly on a vein with a needle, for example, and we can also apply it on our skin like a lotion. Then we have D for distribution, which is how the medicine will get to the part of the body that it needs to do its work. Usually this is done via the blood, because from there the farm can reach almost every part of the body. Once the job is done, we need to metabolize the molecule. This is break it down in smaller pieces so we can excrete it without problem, and it can leave our body without making any damage or accumulating in our organs, which can cause toxicity. And then we need to excrete it. This is how the drug will leave our body. Here we have 4 letters, but it is usually referred as 5. What is the T then? What does the T stand for? T is for toxicity. Toxicity is what we call when we have an adverse reaction or some damage that occurs, and it can happen in any part of the body, in every tissue. Depending on where it affects the toxicity or the damage is done, we can call about hepatotoxicity, for example, if it's done in the liver, cardiotoxicity if it happens at the heart, and even we can talk about genotoxicity if the molecule is harmful for our DNA or our genetic code. The most common types of toxicity, which are the ones in which we focus in our investigation, are clinical toxicity. This is an umbrella term to refer any adverse effects on the overall human health that may happen during clinical trials of a drug before it reaching the market. Then we have cardiotoxicity, which can cause, for example, arrhythmias and other heart problems, for example, by blocking the HERG channels, which are very important for the heart to pump our blood. Hepatotoxicity is very important, a very important kind of toxicity, because of what I said earlier, that most drugs are metabolized in the liver, in order to leave our body. So if the drug is discomposed in toxical parts or has some toxic effects, it can affect directly our liver and produce some injury, drug-induced liver injury, which is called DILI. This happens, for example, with alcohol, which is also metabolized in the liver because it is responsible for usually detoxifying these kind of substances that are external to the body. And we also have carcinogenicity, which refers to the development of cancer. This usually happens by the means of mutations, because there are some compounds that are more likely to cause mutations, or DNA damage that may damage our cells and provoke a tumor like cancer. To test the drugs for toxicity, traditionally it is used animal models. Since there are a lot of animals that share a lot of mechanisms, are tissues and organs with us, and we share many similarities, usually the drugs are tried first on these animals close to us, like mammals, like a rat or pig, and that way we can be kind of sure that if the drug has an adverse effect on these animals, probably will have it also on us. And if it works well on them and doesn't cause them any harm, we need to check in a human example if it's also safe for us or not. Summing up all this process that I know is quite long, we have this diagram. First, we need to find our therapeutic target, that is the protein that we want to attack in order to solve our disease or our problem. Once we have identified our protein, we need to find a molecule from all the possible combinations of molecules that could ever exist that interacts in the way that we want with that protein to a notable effect. This is the lead generation. Then we enter a phase of lead optimization. Once we have identified which molecule we want and which molecule it can be used to solve the problem that we have, we need to optimize this molecule in order to maximize its effect and minimize its possible harmful effects. This is where toxicity is seen. Then, if we are quite confident with our molecule, it can go enter the preclinical phase, which is when we try our molecule on animal models. If everything goes all right there, we can pass to the clinical phase and conduct trials on humans. And if everything goes okay and no problem happens, then we can have our drug approved and it can reach the market and the client and the patients. Overall this process, the number of molecules that reach to the next phase is constantly reduced. And only a very few molecules reach to the final end. In fact, here we have some statistics about this. The 90% of candidate molecules that start the process never reach to the end. And the 30% of these molecules, which is a quite substantial percentage, is due to toxicity or adverse effects of the drugs. 70% of them fail already in the preclinical phase when testing on animal models, and 25% of them do it even in the clinical phase in human testing. So what can we do to fix this or to solve this problem? As a computer scientist, we can make a computational model based on AI or machine learning that takes as input our molecule, the one that we want to optimize in order to avoid toxic effects, and predict if that molecule will turn out to be toxic or not in humans. For this, we need to input the molecule in some machine-readable format that the computer can understand. The most common format for representing these drugs is SMILES. SMILES is a string-coded format in which we encode with letters symbolizing the different elements and bonds among them the structure of our small molecule, our drug-like molecule. We also have fingerprints, which are bit vectors in which we encode specific properties of the molecule that we will talk about more later. We can also represent the molecule as a graph directly with the properties for the atoms and properties for the bonds. And we can even use 3D information of the distribution of the atoms in space to input to the model, like a 3D model of the molecule. And we can also generate deep learning representation, more complex representations like embeddings with some self-supervised models like autoencoders. One important part on molecular representation are what we call functional groups. Every molecule, for example, paracetamol, that we all know, have certain groups of atoms that may have certain effects. This is important for the drugs or for the pharmaceuticals because different functional groups or different structural groups or motifs may be present in certain kinds of medicines, like analgesics or other kind of medicines will have different motifs and different functional groups. This is usually what we represent in fingerprints. Fingerprints, as I said earlier, are a big vector of zeros and ones, which actually are encoding this kind of information. They're looking for functional groups in the molecule, and if it is present, it puts the element to 1, and if it's not, if it's absent, it is a 0. That way we can know what kind of functional groups are present in the molecule. Even so, in the graph representation, that we have an atom level graph representation of the molecule, we could generate a higher level graph trying to highlight these structural motifs that may play a part in different effects of the molecule. This is important because some of these functional groups may be related to toxicity. For example, here we have a molecule, acetaminophen or Tylenol, which is an analgesic that has this functional group, OH. But when it's metabolized by the body, by this enzyme or protein, the cytochrome P450, this functional group is changed to this other one that turns out to be toxic to the liver. So this is very important to have into account in order to avoid toxicity. If we know this kind of functional group related to toxicity, we can, having our deep learning model or computational model, make predictions that are explainable and that highlight which part of the molecule specifically is responsible for that toxicity and emit structural alerts of, hey, this molecule is toxic, do not use it. Even so, we can propose alternatives of functional groups that are non-toxic to complete this molecule and avoid toxicity problems in later phase of the drug discovery process. Additionally, if we can do this with many molecules, we can start to discover new functional groups related to different kinds of toxicity. For example, here we have a Venn diagram telling us different functional groups already known that are related, for example, to hepatotoxicity, or that are mutagens, or that produce skin sensitization disease a kind of allergy. And there are also functional groups that can act as many types of toxicity. Related to our work, we are now conducting a review of different methods and approaches that try to predict toxicity. We are also comparing them in a benchmark in order to know which approach obtains the best results and what things that these approaches try work in order to predict toxicity and which doesn't. Our objective with this, as future work of this thesis, would be to enhance molecular representation in order to incorporate biological or chemical knowledge to our representation of the molecule that will help us improve our model predictions of toxicity. The positive impacts of an early toxicity prediction are safer drugs for the patients because drugs will be expected to have less adverse effects. A shortened life cycle, meaning that we will have a more rapid and flexible process that will make farms reach the market sooner, so the solutions to the diseases will be available for the patient sooner. We will also reduce costs because we wouldn't need to do as many experiments or as expensive experiments. We could do these simulations on computer, which are much more cheap, and also avoid failures that already happen and avoid the rework associated with those failures. And it also has some ethical concerns, as we will be minimizing animal testing and also suffering because the compounds that we try on the first place would be expected to be safer not only for humans but also animals. And this is all. Thank you for your attention, and if you have any questions, go ahead.
Thank you, Noel. It was a very, very clear and very visual presentation. Thank you so much.
Thank you.
Now I start the round of questions. I'll go ahead and have a couple of them. And so I'm curious about the fingerprints,
Yes.
the representation. And so this is an accurate representation of a fingerprint, or there are much larger? Because I understand there are many different functional groups, right?
Yes, this is how you call it, a realistic representation of a fingerprint, but of course they are much larger. The big vector usually is maybe 1024, but it works. Many of them works in this way. There are different kinds of fingerprints. One of them work exactly in this way. Mm-hmm. They check for each position a different functional group or structural motif, and if it's present, it's turned up to 1. If not, it's turned up to 0. There are another kind of fingerprints that start from one central atom, and start doing iterations, being able to how many atoms they can reach and how many functional groups they can find in that range, and that they encode this information also in this format. Mm-hmm. So it's quite realistic, but of course they are much larger.
Yeah, and I got also a question about this. Maybe I'm talking nonsense, but I understand that sometimes these molecules have the groups rotated and they function different.
Yes.
How is encoded that information also if these groups are rotated?
In this concrete representation of fingerprints, I don't think so. Okay, because there are also different kind of representations. For example, smiles are a one-dimensional representation because you have all the representation of how the molecule is, but you lack information about the distribution. Yeah. You can input this to an LSTM, for example, or some sequential model, but that is losing topological information that a graph may have. Mm-hmm. Of the connections. You have graphs which are two-dimensional two-dimensional representations. And fingerprints, most fingerprints are also two-dimensional representations because of this. And if you want to account for this kind of isomeric properties in which a functional group is rotated, you would need a three-dimensional representation in order to account for this, because that's why there are some models that input 3D information and some of them obtain better results. There are also some more complex fingerprints that I think they also encode three-dimensional information, but they are not the most common ones. There are very specific fingerprints that have this kind of information.
So I think the better way is to use three-dimensional, but I suppose this very costly.
Yes, you need to be quite selective, maybe, because you need to be careful in how you input the information. Because, for example, you would think that this graph maybe has all the information that it needs for a two-dimensional representation, but sometimes we have seen in our benchmark that these kind of graphs obtain better results because it's already providing the model some expert knowledge about functional groups that are relevant for the prediction, and the model doesn't have to infer it itself. For example, with 3D information, you have the same problem. If you just input coordinates randomly, or not randomly, but raw coordinates to the model, it may be more noisy than useful. So you need to be careful how you input it, but if you input it correctly, of course, 3D information is very useful.
I see. Thank you so much. And also, I understand that you need very, very large data sets for the proteins. Is this a unified effort to have a whole data set, or you need to ask for these data sets to private entities or some sort of that?
There are many data sets available online for this, of molecules. Not so many of toxicity, but there's already a bunch of them compared to other areas where you may have very difficulty to find datasets. Datasets in toxicity are not that large. Some may be larger, but most of them are medium size or small. So many approaches, and one of the ones that obtain the best results, what do is you have usually online enormous databases of different existable or realistic molecules that you don't know if they're toxic or not, but that gives you a lot of information about molecule configuration. So they usually pre-train the models first on these enormous databases of compounds in order to be able to understand the chemistry of the molecules, and then they fine-tune it for a downstream task in toxicity with these smaller datasets. But yes, data availability is not a problem in this field, but to have correctly tagged for toxicity is more complicated. But you have lots of molecules online that you could use in order to do a self-supervised pre-training with no knowledge of toxicity, but knowledge about the chemistry of the molecules.
Okay, no voy a acaparar al hombre. Do you have any more questions? Yeah.
Just another question about the fingerprint.
Yes.
You are saying that you are using several databases from the internet, and these databases, what do you provide? I mean, you can download the fingerprints, or
in most cases, databases contain the SMILES representation. This is quite useful because although it can be used as a one-dimensional representation if you input it in an LSTM, for example, which is a sequential model, what this string is actually encoding is a two-dimensional graph. So datasets from toxicity, for example, if they do not contain any more information, sometimes they do contain more information, but if not, what they have at least is an smiles and a toxicity value. From these smiles, you can compute with Python libraries that are already open and provided for everyone. You can calculate easily the structure of the molecule. And from this structure, or also these Python specific libraries, you can compute certain fingerprints, or at least the most common fingerprints that just look if these kind of functional groups are provided or that do this iterative process. You can calculate it easily with these libraries that already contain functions to do it. If you want to increment the knowledge of the input representation, that is something that you may want. You need to obtain information from other sources. For example, they usually do not contain information about 3D structure, but there are certain methods to calculate it. Some methods take in account like the charge of the molecules, of the atoms, the chemical charge of the atoms. Some others use quantum methods in order to account for electron variability or things like that. What is quite good in this area is that the molecules are molecules. Even if you don't have that information in one data set, you have ways to compute it usually, because it's not like you are dealing with clinical data that you need to have patients and you need to look to real cases of how that clinical case developed. Here you have molecules and different ways of approaching to calculate these properties about the molecules. So even if the dataset doesn't bring this information, maybe you could use another method or use other dataset where you can find it and include it to enhance the representation.
So I just asked because it seems that the fingerprint method you use is very dependent on the vocabulary of functional groups. I mean, if you have a vocabulary of 500 functional groups, it's not the same as if you use 1,000.
Yes, there are different fingerprints, as I said, that tackle different kind of functional groups. It is dependent on which functional groups you are trying to look at. But since they are easily calculated with a function from some library, you can calculate many of them. There are methods or fingerprints that are very generic, that look for example, alcohol groups or aldehyde groups, or very, very common groups that all molecules have. But for example, there's one molecule which is called pharmacophore ERH, I think, that is a fingerprint, I guess, designed by pharmaceuticals and pharmaceutics, that looks to pharmaceutic specific functional groups. And you have different fingerprints that are specific for other areas. Many approaches do not use one unique fingerprint. Instead, they use many fingerprints because they capture different forms of viewing the molecule from different perspectives.
And do you think the fingerprinting is already a solved problem? I mean, you are using fingerprinting that comes from previous research of pharmaceutics. Don't you think you could be able to discover new fingerprinting, or it's a solved problem?
Like traditional fingerprinting could be kind of solved problem. I mean, you could always develop a new fingerprint that considers more functional groups that could trigger better results, but they're quite established. The approaches that we are conducting a review on are graph approaches that take into account different things. Sometimes they use the graph and complement with fingerprints because fingerprints have some expert knowledge in them. But some other approaches just use fingerprints. They just use graphs with no fingerprint, and they maybe use other techniques on the graphs to include this expert knowledge. Fingerprinting, traditional fingerprinting, like bit vectors like this, is quite established, I think, and quite already solved. But some approaches consider, for example, deep learning representations like also a kind of fingerprint, because at the end you are obtaining an embedding, which is also a vector, like a fingerprint, a one-dimensional representation. But instead of ones and zeros, you have a different range of values that are encoding the same, if not more, properties than traditional fingerprints. So I think the interesting part of this is in this kind of deep representations of the molecules, because traditional fingerprints, like you said, are already quite solved and quite established.
I just understood why they are called smiles. There are a lot of smiles there. Any more questions? None of them? Okay, so let's thank again the speaker.